Hello, everyone, and thank you so much for joining me today. We're going to look into understanding different clinical responses and outcomes and how to report clinical findings. So we're going to look into time to failure, equivalency, cluster randomization, continuous and binomial responses in clinical trials. So what we're going to learn in this class is that at the end you're going to be able to explain the different clinical responses, outcomes, and how to report clinical findings. So this is here where we are going to look into different definitions and findings, and we're going to look into, of course, mainly one type of graph which is very interesting to look at, and it helps us in order to look into a time to failure. It means the time where a drug, for example, may fail, but not because of failure of the drug, but because of the fact that we are in patients who are having, again, a disease condition where, unfortunately, they are going to, for example, pass away, but the drug may now help patients to, again, have a better quality of life and obviously to, again, to stay longer alive. So first, let's go over the review of the clinical research and clinical data. So in clinical research, we are using humans, so that we have humans as participants. And the main goals of the clinical trials is to ensure the patient's safety. Then, of course, we would like to determine and understand the effect and value of an intervention in humans. And obviously, the last goal is to find a better and new ways to treat, cure, and improve health outcomes. So we have 5 phases of clinical trials. In phase 1, we are looking into the pharmacology, the pharmacokinetics and pharmacodynamics related to the drug. And then we are going to establish a dose that is safe in humans. This is why in phase 1 we also look into the adverse drug reactions. We may also now have not too many, of course, subjects, but we are going to look into several types of studies with different doses, and we are going to look into the occurrence of adverse drug reactions or any untoward medical occurrence experienced by our patients using the experimental drug. And then we are going to establish a safe dose or maybe I would say a range of doses, so that we'll be able to go to phase 2, where we will test the safe dose in human subjects. So the number of patients in phase 2 will be higher than the number of patients in phase 1. Obviously, for bigger studies, we may have several hundreds of phase 1 human subjects, but usually even up to 20, 30, we can still establish the safe dose, depending again, obviously, to the study, to the type of disease, and the drug itself as well. And then we will switch to now phase 2, because in phase 1 we could establish the safe dose. Now in phase 2 we are going to look into people, human subjects, having a disease condition. So having the disease condition, we are now going to test the safe dose of the drug in different groups. Now, why in different groups? That's because if we are having a range of dose, for example, we are going to look into what is the optimal dose, even the optimal time to give the product. Here we may have a drug that may have some different formulation. Maybe this is a tablet, maybe this is an extended release tablet, maybe this is an injectable solution. So we will also test the efficacy of the drug along with the safety of that drug in human subjects. Now, if we see that the recommended dose or the safe dose is again effective in patients, then we will need to look into more subjects, because they will allow us to have a higher statistical power when we are going to test whether or not the effect of the drug was an accident. So here what we do is that we are going to look into a higher number of patients, and we are going to have a hypothesis. And the hypothesis for this study or this research will be that's a statement, of course, and the statement is our drug is effective in this specific population with this specific disease at this specific dose. And then we are going to test our hypothesis by having several human subjects, and then giving them, we have different groups, each group receiving again a dose, a different dose, and then we may also have a control, or sometimes we may now compare our product, our drug, with an existing drug. This is where we would like to see if our drug is better than the existing drug. Now, here I would like also to talk about another definition, which is non-inferiority. What is that exactly? When we talk about comparing two drugs, one drug may be already on the market, and this is the first-line therapy. And then another company is bringing another drug, and they looked into the studies, and they see that their new drug seems to be better. Is talking about the efficacy better, maybe because of the safety profile. And in this case, in the study, the second company is going to have a study where they will have, again, the first drug being tested in a group of patients, and they will also do exactly the same tests, or the protocol will be the same, and they are going to test their own drug as well. So they are going to look into the same parameters in a population, and then they are going to compare the results or the efficacy of their new drug or experimental drug and the existing or active drug on the market. Now, there may be two case scenarios, or maybe three. One of them is that by going over the study, they will find out that their drug, the new drug, is even worse than the existing drug. So in this case, obviously, they will have to find out other ways to test the drug or to find other indications, but this is, again, it's going to be related to the strategy of that drug and that company. But you have two other case scenarios. One of them is that their new experimental drug shows to be better. It's going to have a better safety profile. It's going to have a better efficacy. And here, based on this, the study and using two drugs, so your drug and the existing drug on the market, you will be able to say that, yes, statistically, our new drug is better than the existing drug on the market. And because of these facts, and you're going to use statistical tests, we're going to talk about different tests that you're going to use, and you're going to be able to compare both drugs, the existing drug on the market and your experimental drug. So this is the best case scenario if you're a second company. But there is another and the last case where you see that your drug doesn't show more efficiency compared with the existing drug in the market. I'm talking about phase 3 here, of course. And then, in this case, you will still continue to do the study, and you're going to compare, using the same protocol, you're going to compare the existing active drug in the market and your new experimental drug. So you continue to have the same number of patients, the same number of, I would say, the same clusters, or the sample of the population, and then you're going to find out that, no, indeed, your drug is not, again, more efficient than the existing drug on the market. Here, what we do is that we are having another way to look into the studies, and here we can explain our study as a non-inferiority study. What does it mean? It means that we are going to say that your drug is not better than the existing drug in the market. However, your drug is not less efficient than the existing drug on the market. So here we talk about non-inferiority. Your drug is not better, but your drug is not worse. So here what we do is that we are going to conduct more studies. And we are going to look into different, I would say, attributes of your drug. We are going to test your drug. We may see that we have less number of adverse drug reactions. We may see even less or lower frequency of the occurrence of different adverse drug reactions related to your studies. And since in your study you are comparing your new drug, experimental drug, and an existing active drug in the market, you will find out, you may find out in your studies that your product is not maybe more efficient than the existing product, however, it is safer. So here again, this is another very good argument in order to show that your drug is better. Then when you go to the market, you may now, again, of course, you may now be having the registration and having the authorization to sell your product on the market, but then you enter into a competition zone with the existing product where you are going to have a market share of that market that is using that therapeutic class of drug. So now you understand why we are doing the non-inferiority studies, and we also do this, of course, in phase 3. Now when we talk about phase 4, this is where our product is already on the market, so we call that the post-marketing studies, where we are going to look into pharmacoepidemiological studies, or obviously safety studies, where we are going to continue to monitor the safety profile of the drug in patients using our drug, which is now not anymore experimental. This is, as I already mentioned, your experimental drug is safer. For example, it's not, it's not inferior compared to the existing drug at that time on the market. So you're on the market, and now this is a drug, and this drug is having a market share, or so it's having the percentage, a percentage of the population or the patient population for that, I would say, type of therapeutic class. So now we need also to understand several factors here, or several definitions. One of them is, again, about the hypothesis testing. Okay. So don't forget that the hypothesis testing is, of course, to identify the primary response variables. So what we are looking at, the primary response variable can be, it's dichotomous. It means it is yes or no. The treatment is a success or it's a failure for a disease condition. Another factor when you're going to do the doing the testing of the hypothesis is to look into the continuous responses. So you're going to see, for example, the blood pressure is going lower. You're going to see if the diameter of cancer cells is lower, so they are shrinking. We're going to look into the quality of life, which is increasing. So this is a continuous response over time. It's not a yes or no. This is a continuation. So we will need to obviously look into the baseline, so the baseline visit before the intervention. We are going to look into different parameters or variables, and then we are going to, for example, give the drug 1 week after the intervention, then 2 weeks after intervention, and then we do it for the next 5 to 10 weeks. So every week we are going to look into the quality of life, looking into, for example, the blood pressure, looking into blood glucose level, depending on what you are looking at as a variable. And we will see over 10 weeks this change in the response to this variable. We can also look into another type of variable, that's a response. This is time to failure or occurrence of a clinical event. Here, for example, we can look into the incidence of death in patients. So we may be having patients who, unfortunately, God forbids, but they are in a, again, terminal phase of a cancer, for example, and they do have a lifespan here related to their disease condition. If these patients are not being treated, because there is no treatment, obviously, because it's not ethical that we don't give any treatment to any person, that may exist, of course, then in this case, we may see, for example, that there is a type of, I would say, continuous response of the body to that disease, and we are going to see, unfortunately, patients who are going to pass away or expire, and this is just a natural cause. However, we can also look into the same population and use our experimental drug, and here we are going to see what the experimental drug may do to our findings. We can also have an existing drug that is helping patients in the terminal phases of a cancer, reducing or decreasing the probability for them to die because of cancer. And then we can now have another group of patients who are going to receive our experimental drug for the same disease condition. And then we are going to compare the probability of these patients in both groups, again, being exposed to die because of the terminal phase of cancer. So here, we are not looking into, I would say, a treatment. We're looking into reducing harm. We're looking into a treatment that is going to give a larger or longer lifespan to patients who are already close to death because of their disease condition, because they are in the terminal phase of a cancer. So now you see, it's very interesting to see how we are going to look into, I would say, those variables, for example. Now, the examples of variables that I can give you is, for example, the total mortality in this case. So we look into total mortality. We are going to have two groups of patients. We know that they are going to experience or undergo a higher rate of mortality because of a disease condition. And then we are going to compare the intervention with two drugs, and we are going to see the mortality rate, and then obviously we are going to calculate the probability for a patient to die because of the disease conditions, but now comparing patients in two groups where each group is being treated by another drug. So one drug would be the existing drug that we use it as a control group, and the second group of patients would be the patients receiving our drug. And then we are going to look into the total mortality. Now we can also look into the incidence of disease. We can look into the adverse effects. We can also look into different laboratory measurements in order to, again, go over different variables. So, as I already mentioned, dichotomous response variables, success or rate, can we increase blood pressure or not? This may be a question, but you can also look into a continuous response. Are we increasing or decreasing the blood pressure over time? So it's not a yes or no. In continuous response variables, this is observing a trend. And obviously, as I already mentioned, we have the time to failure or occurrence of a clinical event, where we look into the total mortality. And of course, it may also include death for specific causes. Now, we have another variable that we call time to failure. This is a primary response variable, which is again occurring with an event. So, for example, death or infection cured or cancer remission or cancer return or myocardial infarction. These are different cases where we look into time to failure. I give an example. For example, we may have a patient having the risk of experiencing a myocardial infarction. Now, this is not happening now. However, we may have a study where we will have a group of patients at risk taking a new drug, and we may have another population at risk taking an existing drug. And then we are going to observe the frequency of the myocardial infarction over 10 years, 15 years, maybe of treatment as well, in these two populations. And then we are going to look into time to failure. It means that if our drug, the experimental drug, is helping patients to experience less, I would say, a whole lower incidence of myocardial infarction, in this case, our drug is efficient because it can now reduce the probability for patients to experience the adverse drug reaction. Sorry, not the adverse drug reaction, but the myocardial infarction anyways. Now, let's look into the proportion of events. So the proportion of events in each group may be compared. It means that now we have two groups of patients, both having, being at risk of myocardial infarction. And now we are going to look into the proportion of events. So what proportion of patients are going to, experience myocardial infarction? And here we look into if, of course, the proportion in each group is different, then if this is informative. Now, sometimes the length of time to reach the occurrence of an outcome is increased or decreased. Here we talk about life table or survival analysis method. So, in a nutshell, we are going to expose or use the treatment. There can be an existing treatment and our new treatment. So, two different treatments. Use it in a population at risk of death, at the risk of infection, at the risk of cancer remission, cancer return, or myocardial infarction, as I already mentioned. And then we are going to observe this population treated with an existing drug and our new drug. And we are going to see the time to the occurrence. And we are going to now compare to see if by using our drug, the experimental drug, patients are going to experience death, but over a longer period of time. So we are now here reducing, I would say, the incidence and their lifespan in their condition or disease condition is now longer. Or we are looking into whether or not after the treatment with our drug, the occurrence of cancer return or cancer remission is lower now because our drug is more effective, more efficient than the existing drug. So this is the explanation of time to failure. If your drug can affect the time to failure and make it longer, in this case your drug is more efficient than the existing drug, because by using your drug you are now delaying failure, you are delaying cancer remission, you are delaying death. And this is one way, as I already explained to you in the previous slides, this is one way to look into the efficacy of a product. Now, in the time to failure, investigators compared the survival curves for the groups. Now, a survival curve is a graph that depicts the probability of surviving or not having an event. Now, we use non-mathematical models that are assumed in regards to the shape of the curve. But sometimes, to estimate sample size, a mathematical model is used, such as the simple model, where the survival curve follows an exponential distribution. This may happen, but not always. This is because when we are looking into the death, for example, and the mechanism of death, we know very well that it may not be always exactly the same. So when we are looking into the death, we are not looking into different mechanisms or the causes, because many times we may not know the exact cause. We know the reason, but what, for example, a cancer, but what that cancer did to cells or to the organism in order to die, then it may not be always exactly the same mechanism. This is why we cannot put all of those different pathways, involved in the occurrence of death, into one basket, and then to say, okay, if patients they died, that's because of the fact that this is for the same reason. Hence, we are going to have an exponential distribution. It doesn't work this way. We can look into the event or the consequence, which is death here. However, we will need to look into this at the, patient by patient, and we don't have usually, this type of exponential, not decay, but increase. Still, we use, obviously, this type of formula that you see here, which is an exponential, of course, distribution for some times when we are having, again, we have to estimate a sample size. Now, we use, for the survival analysis, we use this type of graph. So in the literature, we call that a survival analysis. And this is called, of course, a Kaplan-Meier curves. So what is that exactly? What we do is that we are going to have, imagine, two patient population, and we know that the outcome of their disease condition for them is death. So when you look into this graph, at the bottom you have, on the x-axis, you have the time. So with the time, it may be months. So we know that when we start a study at time 0, we are going to obviously look into our patients, and we will see that some of them, they are dying at some time because of the disease condition. Now you have group 1 and group 2. Imagine that group 1 is drug 1 and group 2 is drug 2. Now, for drug 2, we are going to look into time 0. And we look into the survival rate or percentage. 1 here representing 100%. Imagine that we have 100% patients in group 2 using drug number 2. So now follow me. I will just look into the Kaplan-Meier curve, and I see that, I'm having the survival for me now at the time 0 is 100%, obviously, because no one died. Okay, we have all of our patients living with their disease condition. And then we see that at the time, for example, 15 days after the beginning of the study and our observations, or also giving the drug, we will see that now we are having about 18, 19 patients who died. That's why we will see in the middle of 0 and 1, which is, I would say, half a month, 15 days, we have now only 82, 83 patients left. And then we continue. We see that every month or every 15 days, we will see that patients are again passing away. They are being treated by the treatment or the drug number 2, but the survival rate here is again lower and lower. And obviously, when we see for group 2, after 3 months and a half, we will see that we don't have our patients anymore. And when we would like now to compare this with the drug number 2, we look into group number 2. And we see now that for the survival, if we had also for group 1 receiving drug 1, we had 100 patients, we will see that in this group, we lose about, unfortunately, about maybe 18 patients, but after a month, compared to the group 2 patients, where we lost the same number of patients after 15 days only. So it shows that the drug 1 is increasing the survival rate, is increasing their lifespan, at least for about 15 days for these patients. And this is a clinical event. This is, again, a result. This is a positive result, because for us, we see that those patients, they are living more or surviving more, again, for 15 days. And now when we continue, we will see that even after 1 month, okay, we have the same, but after 2 months, we will see that, again, we reach, I would say, about 63, 64 patients, now the survival rate. But in group 1, that was after a month. But in group 2, this is after, group 1 is after 2 months. In group 2, this is after a month. So now we see that more we continue with the use of our group 1 or drug 1, we will see that we have a higher survival rate based on the time. It's now like we are delaying those patients to die. And if you look into the time of the treatment, more or longer is the time of the treatment, and you can see that on the time now scale. We will see that in group 1 with patients using the drug 1, we even have patients, about 20 or 18 patients, who will survive after 3 months and a half, or even after 5 months. So this is an interesting and a good result, showing that, yes, in fact, if we start, for example, or if we have started sooner the group 1 or the patients using drug 1, obviously up to 20 or 18% of patients could have, again, increased their lifespan within, I would say, or within the conditions or disease conditions that they are experiencing. So this is a survival analysis. We compare the effect of a drug with a placebo, for example, if ethical, but usually 2 drugs, and we are going to see which drug is now increasing the survival rate in patients with a disease condition. And it shows efficacy for a drug. So here in this graph in front of us, we have group 1 that is, again, using a drug number 1, that has higher efficacy when we are looking into the survival curves or the Kaplan-Meier curves. So now let's go over talking about equivalency of interventions. Now, effective interventions may already exist and be standard of care for a disease or medical issue. So we already have a drug on the market, but we are having an experimental drug, and we want to test it. We want to test if whether or not this is better or equivalent to the existing drug in the market. So one of the goals of clinical trials include includes developing and implementing new interventions that may improve treatment or cost less or take up less time or have few side effects and or have less of an impact on the patient's quality of life. So pharmaceutical companies usually, one for example may have one treatment, that's what we saw in the last slide. For example, that was drug 1 or drug 2, here we call them drug X and Y, and pharmaceutical company 2 is developing a new treatment like drug Y. Now in the scenario we have a clinical trial with a positive control. The difference in events rates can be P1 and PC in some values delta. Now therefore investigators can specify a value of delta equal to X, and values less than X are considered equally effective. This is like we are saying that the existing treatment is, for example, efficient at 30% in 30% of patients, and now we are going to look into whether or not our drug has the efficacy which is higher than 30%, so it's 40% or 50%. This is what this explanation, or say this is what how we can explain what we just read together. Now here, let's reflect about that. It's a cancer equivalency study. So it's a study that tests whether a new treatment is not worse than an active treatment it's being compared to. So this is what we call a non-inferiority trial. So a non-inferiority trials are sometimes done when a placebo, an active treatment cannot be used. So here what you see is that we have two drugs. We have levantinib and we have sorafenib. Now sorafenib is in red and levantinib is in blue. So what we can see is that we are looking into the Kaplan-Meier curves and analysis for overall survival with stratification factors. Okay. So on the left side of the this graph, we can see on the y-axis the probability of survival. So at the beginning of the study time 0 months, at the time 0, we see that the probability for all of our patients to stay alive is 100%. It's 1. But then we are going to see the number of patients who are going to again die because of their disease conditions over the next 42 months. And we are going to have two groups of patients being treated by two different drugs. And now what you can see here is that this graph is showing that we don't have that much change when we are using any of those products. Obviously, for sorafenib in red, we may see that after 18 months of treatment, we may indeed see a very slight, but still a change in the survival rate or the probability of, again, surviving. So we can now, from this curve, understand that up to month 18, our drug and the, or any of the drugs, so I would say levantinib and sorafenib are both having the same survival rate. However, after 18 months, based on the study that we have, we will see that, again, sorafenib is, again, showing that it has a lower probability. So now here, we can obviously use the graph, and this is one way to show, I would say, and use the Kaplan-Meier curves. Now, in the second case here, for example, we are using levantinib and sorafenib, and we are looking into the Kaplan-Meier curve. But here, this is a different case scenario. Case scenario in a way that we are having a, for the sake of this course, obviously, this is another way that we can look into the treatment. So here the study again tests whether a new treatment is better than an active treatment it is being compared to. And what we can see is that yes, in fact, when we look into the probability of again survival, we will see that if our patients are using levatinib, for example, the curve is higher, so there is a higher probability of survival compared with sorafenib, where this is, we have less probability of survival. So what it shows that this is, again, levatinib is, again, offering a better condition for the survival for the patients, because there is a higher probability, especially after a month, I would say one, for patients to survive. So now let's talk about the confidence intervals in clinical trials. Confidence intervals, they provide a measure of the reliability of an estimate from the data. Now, you may ask, how reliable is the data? Obviously, this is where we talk about confidence interval. Now, the confidence interval is the mean of your estimate plus and minus the variation in the estimate. It's like your average plus or minus a value. So therefore, we call it, of course, the standard deviation, but now, therefore, it is the range of values you expect your estimate to fall between a certain percentage of the time. Now, the confidence level is expressed as percentage, and is the percentage of times you expect to reproduce an estimate between the upper and lower bounds of the confidence interval. For example, upper and lower confidence limits of 95% indicates that it is likely to be the same 19 out of 20 times if repeated. I will explain this in an easier way, is that when you have your lower confidence limit at 95%, it shows that up to 95% of times when you use your product, it is working. It means that only 5% of time, it's 100 minus 95%, so it's a percent is 100. 5% of time, you may not see an effect of your drug that may show some efficiency. What does it mean is that now our confidence interval is 95%. It means that we know, we are confident that our drug is working at 95% of cases. So out of 100 patients, 95 patients are going to benefit from the drug. This is what we call a confidence interval. So now we have also another term that we have to go over, which is the cluster randomization in clinical trials. Why we do randomization? That's because we cannot choose our patients. We cannot choose them based on what we know about them, how we see them, based on their, I will say, different attributes that they have. Everything must be done in a randomized way, so choosing them by hazard, so that when we have our study participants in different groups, all of those participants have been chosen by hazard. We didn't choose them to say you must all have a specific height or specific age or specific, I would say weight, and they are going to be randomized. Now, when we talk about cluster, this means that we are going to choose our, again, study population from a specific population, but still randomized. For example, we would like to have a study of, again, some intervention in a school. And in this case, we are going to choose only students within that school. It doesn't mean that we are going to choose exactly the same students or look choosing them. It means that within the cluster of that school, we are going to choose our students. This is what we call a cluster randomization, because now if you're having 200 schools in your area, we are only going to choose randomly the schools. So we have school 1 through 200, and randomly we are going to, we will see that school 1, 10, 25, 64, for example, are randomly chosen. This is a cluster randomization. And since this is testing, again, an intervention in humans, this is a clinical trial. Now, let me give you an example. So, for example, investigators might be interested in understanding if a smoking prevention strategy at school can work. So here, the investigator may randomize the schools where some schools get the new smoking prevention strategy and others do not. Obviously, if this is ethical as well. So each school is a cluster, and the study undergoes cluster randomization. Now let's go over the cluster randomization that can be now divided into two continuous or binomial responses. So continuous response variable is the response which is measured on each individual within the cluster, so within each school in this case. Now, variability of response is due to differences in individuals with a cluster plus within the cluster plus differences of individuals between clusters. So this variability can be measured by interclass correlation coefficient P, which ranges from 0 to 1. Now, intra-class correlation can vary in clinical trials from 0.1 to 0.4. Now, we can also talk about, again, continuing with the binomial response. Again, this is a measure of degree of within cluster dependency or concordancy rate in participant response. Now, typically, investigators use the kappa coefficient K. So a concordant cluster within K equal to 1 means that all responses within the cluster are identical, either all success or all failure. So this is again a way that we can use the cluster randomization and use the data and to conclude from the data. Now we can also have multiple response variables. So clinical trials occasionally have more than one primary question and more than one primary response variable. So we may have now multiple responses that can arise if investigators cannot agree about which factor is the most important, because these are multifactorial responses. So multiple responses are required to assess a single primary question. However, the multiple responses and comparisons are made, there is a greater chance to find significance simply by chance. Obviously, we talk about the chance, but mainly in statistics, we talk about the probability or likelihood. So we may observe, of course, multiple responses. It means that we are having more variables involved, which is normal. We are in nature. This is natural that we are having several factors, also confounding factors that will affect, the results of the studies that we do. But what happens here is that since we are dealing with so many factors, we use the probability. We use tests in order to measure the likelihood, the way to measure and explain the likelihoods for a specific sample size, obviously, for a specific disease condition at a specific time, in order to explain, of course, those different response variables. And obviously, through those statistical analysis, we will be able to look into whether or not the effect of an intervention is just an accident or based on the confidence interval. When we are using a drug or an intervention, we know that up to 90-95% of our patient population is going to benefit from that intervention. So I want to thank you so much for joining me today. I hope that you enjoyed today's lecture. Thank you, stay well all, and see you next time. Bye-bye.